Hey team, Dr. J Cordy here, and I'm going to take you through in this video, what does SARS-CoV2 do once it's inside your cell? So in the last video, we went through how does this coronavirus get into the cell, and now we're going to go through what does it do once it's there. All right, let's jump into it. So here's the rough life cycle of the cell, and in the last video, we covered this part, and now we're going to cover this part. What happens once that genome is inserted into the cell? Because remember, it's not really the virus gets into the cell, it's that the genome of the virus gets inserted into the cell. What does that genome do once it's there? So let's have a quick look at the genome. Now these are some cool proteins I'd love to talk to you about, but we've got to focus. I tend to get distracted. Let's have a look at these blue ones here. One's called replicase and the other one's called helicase. Replicase and helicase. Now, replicase goes by a couple of other names. One is RNA-dependent RNA polymerase, and the other one is just RNA polymerase. I prefer to use RNA polymerase because that's what it is. It's an RNA polymerase. It's just not our RNA polymerase. And that's the key point. What we can see here in the genome of the virus is the machinery to duplicate itself. It's one of the very few things the virus does not steal from the host cell. It actually generates the machinery to duplicate. It actually encodes, it has the codes for the machinery to duplicate its own genome. Important point. Right, so here's an abbreviated version of the genome. It's not the full genome, obviously. It's just an example. So here we have a bit of RNA genome. So this is the viral genome. What happens first? Well, the first thing that actually happens is it gets translated into proteins. And that's because we need two of those proteins. So the first thing happens is the cell's ribosome, so that's our ribosome, comes along, takes the RNA and translates it into protein. And two proteins come out that are critical for what's about to happen. RNA polymerase, replicase, and helicase. Now, I always think of helicase as a helicopter because what helicase does is it chops double-stranded nucleotide molecules into single-stranded. And I always imagine those rotor blades coming along and separating the double strand into two single strands. So these two proteins have to be produced before the genome can be replicated. So the cell's own ribosome does this, and it uses its own amino acids to create these two proteins, which are essential for the genome replication process. So now we need this RNA polymerase to do its thing. So here we have the SARS-CoV2 genome, and the RNA polymerase comes along, and what it does is it takes the sense strand and it makes a complementary strand. So wherever there was an A, there is now a U. Where there was a C, there is now a G. A lot like our DNA. But now we've got a double-stranded RNA molecule, a sense strand and a complementary strand that is sometimes called the antisense strand. Now, there's nothing we can do with a double-stranded RNA molecule, so what we need to do is chop that in two, and that's where helicase comes in. So the rotor blades of helicase, which is actually a protein, it does not contain rotor blades, it chops the double-stranded RNA into two single strands. Remember, one is called the sense strand, and the other one's called the antisense strand. Now, it's no good creating a whole bunch of antisense strand. It doesn't code the proteins, and it's not the genome of the virus. There is almost no point in having it. It's basically useless, except for the one thing that it does, is it provides a template for the sense strand to be created from. So what's going to happen next is the RNA polymerase is now going to go over and over and over again on that antisense strand, creating a complementary strand to the antisense strand, which is the sense strand. So now if we look at this, it goes blue, orange, green, green, blue, orange, green, green, blue, orange, green, green, blue, orange, green, green. We have effectively replicated the RNA genome, and this can happen hundreds or thousands of times inside a single cell. So the RNA polymerase, which was encoded in the SARS-CoV2 genome, but it was created from the cell's ribosomes and amino acids, then goes on to replicate that genome, the antisense strand of the genome, over and over again to give us multiple copies of the sense strand of the genome. Right, So now we've got the replication done. An important point is that the RNA polymerase replicates the whole genome a lot. So we end up with loads and loads and loads of genome ready to go make new viruses and spread, but it also creates just genes, basically genes, but they're called subgenomic transcripts. And basically they're smaller pieces of RNA that encode a specific protein. So they're just like our versions of genes. And so the viral genome just replicates small chunks that can then go on to ribosomes to create the proteins for the virus. So the whole genome gets replicated as well as just the genes of the virus, or just subgenomic transcripts that go on to encode protein. So here is the nucleocapsid protein, that's the protein that helps package the RNA, the spike protein, which recognizes the ACE2 enzyme, membrane and envelope proteins, which help form the structure of the virus. And that's where we're heading to next. Right, so we've covered this genome replication part of the SARS-CoV-2 life cycle. Now let's cover how does a viral particle form and then get out of the cell. So this is actually straight back to basic biology, because now the subgenomic transcripts are just going to get plugged into the cellular machinery, right? So the subgenomic transcripts are going to go into a ribosome. At the tail of each of these transcripts is a signal peptide, right? So it is a peptide that tells the ribosome to go to the rough endoplasmic reticulum, because this is either to go inside the lumen of the rough endoplasmic reticulum or to get stuck halfway and become a transmembrane protein. That's recognized by the signal. That signal peptide is recognized and it signals for the ribosome. I don't want to overcomplicate it. It signals for the ribosome to go to the rough endoplasmic reticulum. So now the viral proteins can be created straight into either the lumen of the ER, so either inside the ER, or to get stuck and become transmembrane proteins. So the ribosome here, this is a picture sort of a whole cell. The ribosome picks up the subgenomic mRNA transcripts from the viral genome, recognizes that it's got a signal peptide, so it should go to the rough endoplasmic reticulum. It joins the rough endoplasmic reticulum. The proteins end up inside the rough endoplasmic reticulum lumen, or stuck inside the membrane. This then buds off into a vesicle and heads towards the Golgi. Now, it doesn't actually go all the way through the Golgi. It gets stuck in a structure that's called the Golgi intermediate compartment. And this is where the viral particle forms. It doesn't want to go through all the Golgi, and so it takes a little shortcut. It goes from the rough endoplasmic reticulum to the Golgi intermediate compartment. And here we can see all the proteins are stuck inside the membrane. These are membrane-bound proteins, and in green we've got the spike proteins. Now, what's going to happen, and this is actually super, super cool, the proteins are bound in the membrane, and you can imagine, this is a little bit simplistic, but you can imagine they have almost a curved structure while they're in the membrane. And if we end up with these proteins with loads and loads of curved structures embedded in the membrane, they actually bend the membrane round until it forms a full circle. Now, the full genome transcript, not the subgenomic transcripts, but the full genome is then bound to the nucleoproteins to form its sort of like densely packed genome plus protein. And it is then attracted to these membrane proteins that have been produced by the viral subgenomic transcripts. And what happens is the curved proteins that are bound to the membrane then create an entire ball of the phospholipid around the genome, right? And so because it's kind of stretching this genome around here, but this part of the phospholipid membrane remains not part of the viral particle, and it just remains part of the Golgi intermediate compartment, we end up with a viral particle inside a vesicle, right? So we end up with a full viral particle that's formed from the membrane of this Golgi intermediate compartment. I wish there was a smaller way to say that. But basically, this membrane compartment gets bent around to create the viral particle, and a whole nother layer is bent around to encapsulate that viral particle. This then actually goes through its normal life cycle. This is often what happens to vesicles that have come from this ERGIC or the Golgi. They go to fuse with the membrane and release their contents, right? Because this is what often happens to proteins that go through the Golgi. They are secreted, or they become part of membrane, or they become the membrane proteins. So the viral particle then is released into the environment, which is amazing. It is totally stealing the machinery of the cell in order to create viral particles that are then released. Very cool stuff. Oh yeah, so in the next video, we're going to go through a technique to detect whether you've got the virus. How do we know you've got the virus? And that will be coming up in the next video.